Thank you so much. First of all, I want to to acknowledge the organizing committee for giving me this opportunity to present some of the work we've been doing in the lab and to give you a hint on what are our biofilms and biofilm-related infection. It's a great honor for me to be here and opening this session about AMR. Okay, so I cannot start something about AMR without showing you this picture. Maybe some of you know already this. This is something related to the number of deaths by disease and other type of problem like a road accident. We are here at the moment about 700,000 deaths by infection, and because of AMR, the prediction are really bad, and that in 2050 we'll be reaching probably 10 million deaths a year. These are the classical mechanisms of antibiotic resistance that have been discovered and now are super well known and well described. I won't go through all this, but this is something related to the entrance or efflux or influx of the antibiotic, the modification or target amplification, the repair of the target, etc. And there are multiple mechanisms that have been described. What I'm here today is to try to convince you that there is another way bacteria have found to overcome killing by antibiotics, and this is making biofilms. And that these biofilms, they should not be neglected when thinking about fighting infectious disease. So, for a long time, we've been thinking that bacteria and other microorganisms, by the way, were growing as isolated microorganisms. And they were, here you can see bacteria that are swimming on the liquid. We know now that bacteria can live in community, and bacteria and also other type of microorganisms that can live in community that are called biofilms, and that these bacteria can, switch from one state to another, sometimes moving to the planktonic state, and then try to colonize other niches, coming back then to the biofilm lifestyle. So this is what you will find in books about what is the biofilm formation pathways or development that start by an adhesion of the bacteria to the surface. Then the bacteria will eventually start to attach and swarm on the surface, will develop some other bacteria creating some what we call multi-species biofilm will eventually enter into the biofilm, and then at one stage they're going to start producing what we will see polysaccharides, and then they will produce a major biofilm that can eventually be dispersed either by pieces of biofilm that will be sloughing out, or a bacteria that will be reactivated for swimming, for example, and then go again in this cycle. You see that this is a question of time scale. We can go from seconds to days, years on this system. So if we want to give a definition of biofilms, I would say something like a community of microorganisms that interact with the surface and that are embedded in a self-produced matrix. This is the basic definition you will also find in books that can be adapted in function of, say, the situation that you're facing. These are examples of what we can call biofilm in the life, in real life. So colony on plates, some people are considering that colony on plates are biofilms. Microbial mats that can be sometimes very thick, like several centimeters, even meters in some cases. The dental plaque is a biofilm, for example. And then you will find also biofilms in the body, like in the lung, in the intestinal lumen, and then also in the environment, like as bacterial aggregates. Or wastewater sludge are also considered as biofilm. Okay. We will come back to that, but when biofilms form in implants, for example, this will be a major concern and problem. So biofilms are everywhere, okay? They are growing in mineral surfaces, vegetal surfaces, even microbiota, mucosa, etc. And then bacteria switch from one style to another. And then finally, they have been forgotten for a long time. I like to present this slide that is a sort of an history of the biofilm. In fact, the first observation of a biofilms dates from these dates, so 1684, by a guy that was called Anthony van Leeuwenhoek. I probably didn't give the right name. You'll be better telling exactly, so I'm not super good for the name of Dutch people. So which was the guy that discovered the first microscope, okay? And when he would do that, he would observe his plaque. You know, he was scratching his teeth and was putting that under the microscope. And this was the first observation of a biofilm, okay? So it was a long time ago, and it took like more than 200 years before we start to see other experiments on biofilms by Louis Pasteur that was working on aggregates involved in acetic acid fermentation. And then only, again, a lot of time comes before a guy called Arthur Henrici looked at biofilms under environmental surfaces. And then, during that time, biofilms were called slim, biological films, glycocalyx, biofouling, and the term biofilm has only been coined in 1975, okay, by a guy called Mack. And in fact, this guy was in a group of this person, that is Bill Costerton, that is the father of biofilms, I would say. He actually, unfortunately, passed away a few years ago, but he has been the guy, together with Niels Oeby, that actually pushed the field of biofilms. And in fact, there is a link between the fact that biofilms are now studied a lot and, in fact, the fact that biofilms have been discovered, been involved in clinical infections. The first observation of a biofilm related to clinic comes from this work of Niels Oeby about Pseudomonas aeruginosa that was infecting, in patients that suffered from cystic fibrosis. Then this has been published later in the 80s. And then this is the first observation of a biofilm on a medical device. Okay, not observation, but at least images that we can have from the work of Marie et al. Again, people from Bill Costerton lab. And this was here a colonization of a catheter, a pacemaker by SRS. So maybe now you know that finally biofilms in the clinic, human clinic, okay, they come in two flavors. One is what we call tissue-related infections, and then can cause also device-related infections. So in red here, you have all the diseases that we have identified, and they are maybe not here, all here, that are actually linked to the presence of a biofilm. And on the left side, this is all the device-related infections that have been detected so far. Okay. If you look to the literature, you will find that 60 to 80% of the human infection have a link with a biofilm. That is actually huge. Okay. So I'm not sure that these numbers are true, but what can I tell you is that you should think about a biofilm in terms of infection if you are if you are working in a human clinic, but also if you are working in the veterinary clinic. So just a few numbers, for example, for device-related infections. So there are huge numbers of infections, a number of devices that are actually put, here it's in the US, like, very important. And what you can see is the prevalence rate of infections of some of these devices is super high, like almost between 10 to 30%, for example, for urinary catheters. It's also the case for venous catheters, also with some percentage that can be very high, eventually for hemodialysis catheters. And I will come back to this type of catheter because we use them as a model to build an animal model later. So high numbers of insertion of device in the body and a high rate of infection. So if we come to something that is more fundamental about what are the main characteristics of biofilm, I would say that one of the main characteristics of biofilm is heterogeneity. Why in a planktonic or batch population you will more or less find bacteria that are facing the same environment and have approximately, would say, this is maybe not absolutely true, but the same phenotypes. In a biofilm, you will find a zoo in terms of heterogeneity, of expression of genes, of phenotype. And the reason is this, the biofilm matrix that is actually produced by the bacteria themselves, that in the body will also eventually be using some of the host protein and host polysaccharides, and that are made by water, protein, extracellular DNA, and polysaccharides. Because of this matrix, you can imagine that a bacteria that will be here and a bacteria that will be here, they won't face the same environment. You have 3D architecture and then gradients of oxygen, nutrients, and waste. And because of that, then you have micro-niches where bacteria will face different type of environment. And then this explains the heterogeneity. Because of this, you have possibility of local differentiation and differential expression of genes. And then this we expect having biofilm-associated functions that are different from the ones that are found in the free-living bacteria. So what are the consequences of this? Okay. So the first consequences is that these bacteria, when they are in the environment, so I put medical surface, but this is true for any type of environment, they have an increased resistance to a lot of different things, including desiccation, mechanical force, nutritional deprivation, disinfectant. They're also more persistent in the host, with an increased resistance to the immune system, with reduced penetration of the immune cells, IgG, and the stimulation of apoptosis. And for the list, last but not least, increased tolerance to antimicrobials. That is the subject of today. Okay, it is actually accepted that basically biofilm will be 100 times to 1,000 times more tolerant than planktonic free-living bacteria when facing antimicrobials. And then they are very difficult to eradicate. And then this is again funny to see that our good friend Anton at that time, he already observed that, because when he was saying that from when I'm conclude that the vinegar, so he was using vinegar, that was not antibiotic at that time, but he was saying that when he was using vinegar, he couldn't remove all the bacteria that he had in the mouth, and that the last layers of the bacteria were still alive. And this is exactly what's happening now when you do use an antibiotic against a biofilm. So how you can illustrate that if you have, a biofilm in a clinic, how you can eventually suspect that there is a biofilm infection? I would give you like something very simple. If you have an animal or a human that is sick, has fever, then you would eventually do an infection. You would sample the blood or the site of infection, and generally you will try to isolate the bacteria, potentially if they are not virus, because if they are virus, this is something else, but if they are bacteria, you will isolate the pathogenic bacteria, and then you would do a classical antibiotic susceptibility assay to find out this sort of thing where you're going to find an antibiotic for which the bacteria is susceptible. Then you say, okay, I'm going to use this antibiotic in my, for my patient or my animal. You use this antibiotic at a very high concentration, and what's happening? Either nothing happened, so you have a problem because this antibiotic apparently has no effect, or eventually, and which is most of the case, you will see that the fever will get out, the infection site will be cleared for some time at least, and then few days from weeks later, then it's coming back. And this is classical of chronic infections, and this is always then a suspicion of a biofilm-related infection. Okay. So what do we know about the mechanism of increased tolerance to antimicrobials? So there's, in function of the bugs, you will find some specific mechanism in induction, and the one that is the more known at the moment and described is an efflux pump. So pump that will pump out actually the antibiotic outside the bacteria, and these are actually activated specifically at a time where the bacteria are within biofilm. A very important aspect of biofilm tolerance, and is again linked to this matrix, is the reduced penetration of the antibiotic within the matrix, through the matrix. So the matrix in general, you will find about 40% of reduction of diffusion. That means that for a certain time, the bacteria, when the antibiotic is reaching, the bacteria will face a concentration of antibiotic that is not enough strong, that is not reaching the MIC, and then they will be inducing somehow some mechanism because they have little concentration of the antibiotic only, and then they can respond much better to the antibiotic. There is also concentration of active enzymes like beta-lactamase or chrome acetyltransferase. There is also something that is genetics, like would include increase of the genetic mutation rate, and then this is often due to stress response, and this I won't go into this detail. You may know that also in biofilm there is induction of horizontal transfer, and this is a main concern in terms of dissemination of antibiotic resistance. And this is important for two reasons in biofilm. First, you can directly transfer resistant genes that are carrying by plasmid from one bacteria to another bacteria. And this is the most known example. The other one is somehow indirect, because these plasmids, they carry often adhesion factors. If this plasmid is transferred, the bacteria that will receive it, it will be doing much more biofilm, and then that's a cycle, more biofilm, more tolerance, more transfer, more biofilm, more tolerance, more transfer. And this is a bad cycle for the patient in that case. One thing that is now super important in the biofilm field is that in this biofilm, there is an increased frequency of what is called persister cells. Alors, persister cells are cells that are a bit specific, and I will give you just a little bit of information about those, but they are finally what we think are responsible for chronicity of disease. In that case, if you have the persister cell in the biofilm, antibiotics will eventually, together with the host immune system, kill what is outside, and the antibiotic will kill what is inside, but the persister cell will survive the antibiotic until when you're going to stop, because you're going to stop the antibiotic because the patient feels okay or the animal feels better. And then when you stop the antibiotic, then these bacteria will start to divide again, and they will reinfect the host. And this is then a reason of recurrence. So these persisters are the main cause of the recurrence chronicity of biofilm resistance related infection. They are more abundant in biofilm than in planktonic population. It's a transient phenotype. It means that if you grow this cell, you treat, there will be here the persisters. If you remove the antibiotic, they will regrow. But if you treat, you'll have more or less the same frequency of persisters, and they might not be the same cell. Okay, so persisters is it's a phenotypic test, that phenotypic presence, and it's somehow a bit stochastic. These persisters, they can pre-exist before antibiotic, or they can be induced. There are of two types. And what is somehow very important is this. A resistant mutant will grow in presence of the antibiotic. A persistent bacteria will not grow. You can kill most of the population using the antibiotic, and then you will remain with this bacteria that you can increase the concentration of antibiotic, you will never kill them. So they have been described as somehow dormant cells. Okay? So they are difficult to manage because they are not growing. And a lot of antibiotics are targeting growing bacteria. Okay, and there is something that is very important to the topic of today, is that there is a link between biofilm persistence and emergence of resistance. Okay? First of all, because of the increased duration of residence of the bacteria in the body, this enhances the odds for resistance to emerge. That's the first aspect. The second aspect is that because bacteria from biofilms face stressful conditions, you'll have an acceleration of the mutagenesis and the horizontal gene transfer, and that we know these mechanisms are promoting resistance. And also what has been shown is that the stress response that are promoting persistence are in fact more or less the same stress response that are promoting emergence of resistance. So biofilm persisters may represent a pool, a reservoir of organisms from which resistant mutants can emerge. So we better trying to fight biofilm to also avoid emergence of resistance. So about fighting biofilm, okay? What type of techniques or strategy have been developed the last, I would say, 20 years in the field? So this is the same development that you've seen. So I will come back to the different type of strategy, etc. But I will show you one or two examples of some strategy that we developed in our lab. And the first one is linked to the first step of adhesion. And this is true for an adhesion to a medical surface, but also to any mucosa. So what we've been doing in our lab, for example, is to use biomimetic approach to globally inhibit bacterial adhesion, and notably use glycocalyx like sugar or molecule that are non-biocidal anti-adhesive compounds, and this is a very important aspect of it. So we've been showing, for example, that natural hydrophilic polysaccharides could be used as anti-adhesive molecules that can be grafted to medical surfaces and then avoid bacteria to attach. But these molecules are very complex, and you cannot produce that at a very large quantities. So, in fact, having worked with a chemist at the Institut Curie, we've identified a simple hydrophilic molecule, and one of it is methylcellulose that you can produce at very high quantities. It's an FDA-approved molecule. And then we've shown that this has an anti-adhesive properties in vitro. It's not toxic to a eukaryotic cell, and it's not killing bacteria. It's not killing bacteria. Question is, how can we use that in a real medical device? And because you have the presence of this host cell and proteins, and that this will not be super good to use this this methylcellulose in vivo. So to develop a model for in vivo test of this molecule, we focused on one special device that is the totally implanted venous access catheter, or TIVAP, that are put in patients that need to follow cancer chemotherapy, or parenteral nutrition, or cystic fibrosis chemotherapy. So there are chambers that are put directly under the skin, and that can be accessible by crossing the skin barrier, and then with a catheter that is directly inserted in the jugular vein and goes in the heart. And this is used, to put chemotherapy directly in the body. Problem: this is in fact in 3 to 6% of the case, and then this is a major problem that you need to remove the catheter to treat the patient. So we've used this with a rat model. So we used a pediatric catheter that we can implant in the back of the rat. We can insert this in a jugular vein of the rat, and this is the scar at the end with here the chamber in the back. It's possible to inoculate bioluminescent bacteria and follow the development of biofilm over time using a CCD camera. You can see the development of the biofilm. This is the back with you see here the chamber, and here the catheter that goes then on the jugular vein. You can maintain this for more than 150 days, for example, mimicking chronic infections. And then you can get the catheter out. You see the biofilm inside the catheter. You can actually count the bacteria. So we've been doing that with different bugs like E. coli, Pseudomonas, Staph aureus, Staph epidermidis. And you can also visualize the biofilm within the catheter, and you can see very super nice biofilm in this. So using this methylcellulose, what we did, we disassembled this TIVAP. So you have few pieces. There are some pieces here that are in silicone, and the port here is actually in titanium. And then we've been grafting this methylcellulose on the catheter, and we've been using this in our model. So you can see here that this is untreated. So when you put S. aureus here, you can see the biofilm is developing nicely. If you put silicone grafted or silicone plus titanium grafted, you can see that you have a reduction of the luminescence. And here it's counting of bacteria for 2 bugs. And you can definitely see that if you graft with silicone or silicone plus titanium, you have something like a 4-log reduction in the bacterial colonization, which is a super high effect from what we've been seeing in the literature. What is also important is that these molecules, they also are antithrombotic. So often these catheters, when colonized, they will be just clogged. You can't use them anymore. You have to remove them. So this molecule also avoids clogging. Here, if you have a treatment, you'll see you have a 22% clogging. If you have no treatment, you have more than 90% clogging. So this molecule not only reduce binding of the bacteria, but also reduce binding of host cells and protein, so which is very important for a molecule that could be used in the clinic. Okay, so the first take-home message that would be, if you want to inhibit initial adhesion, you can use non-biocellular anti-adhesive molecule. They can be of broad range activity, like the one I just presented. You can also develop some molecule that will target specific adhesins of the bugs that you're working with, and then eventually you will inhibit only that bacteria and not the other. That can be sometimes important in some application. So what is important here is that you will limit the toxicity, and more importantly, you reduce the risk of resistance, because you're not killing the bacteria, you're just pushing it away, and then so that the immune system, for example, will be able to handle it. So the other strategy I just want to point is the reduction of biofilm tolerance. And then I will not give you all this information because I could speak for hours about that. But these last years, there's been a lot of strategy that have been developed to kill what I described just before, the persister cells, these dormant cells, okay? And there are multiple strategies that have been used, and if you're interested, we can talk later about that. What I just going to present you today is also a way of killing persisters, is to actually potentiate the activity of the antibiotics. And then we can use different type of adjuvants that will potentiate this activity. In our case, what we were thinking about was to try to facilitating the dispersion of bacteria from the biofilm will help the antibiotic to kill the bacteria when dislodged from the biofilm. And we use the property of an agent that is called EDTA, that is a chelating agent. So it's a very simple molecule, and that actually removes the divalent cations from the biofilm, and this is destabilizing the matrix. And there's also another property of this molecule. In fact, long time ago, people have been showing that if you increase pH, you increase the efficacy of aminoglycoside to enter into the bacteria, and then you decrease resistance to aminoglycoside. So EDTA is alkaline. So then you have two effects. One effect is chelation and then destabilization of the matrix. The second effect is increasing pH so that you will reduce resistance to aminoglycoside. So now testing that in vivo. How we did this? We did this by what we call antibiotic lock therapy. And some of you may know this sort of technique that are actually used in clinic to save the catheter sometime when they are infected. So this is a local treatment where you're going to inject into the chamber a very high concentration of the antibiotic, a very little concentration, a very little volume, but high concentration, and you're going to leave that for 12 to 24 hours, and you're going to renew this antibiotic lock every day. And generally, it is used for something like 7 days before you try to eventually see the effect of this treatment. The problem is that it cannot be used for bacteria that are called, like Pseudomonas aeruginosa or S. aureus, and the clinic recommendation is always to remove. And even for bacteria that are treated by this, the success rate is not so good, 50 to 80%. And then you're ending often to the situation where you have to remove this catheter anyway. So we use this catheter lock system, try to fill the catheter we were manipulating with this antibiotic solution. And this is here the example of our biofilm. In the case, it's E. coli or Pseudomonas aeruginosa, for which we did a 1-day treatment, that is versus 5-day treatment in the clinic. And you see that with no treatment, the biofilm is maintained after the biofilm recover for 7 days here, while the same thing for Pseudomonas, while if you put this mix of gentamicin and EDTA, you apparently have super strong effect on the biofilm. And it's indeed here, put also in numbers, E. coli, Pseudomonas, when you treat with gentamicin alone at super high concentration, so you have a good reduction, but you still have this persister cell that stay in the biofilm. If you mix genta plus EDTA you see nothing that is detectable. So in fact, we have an eradication of the biofilm with this mix, and this was shown to be true for these two bugs, but also S. epidermidis, methicillin-sensitive S. aureus, methicillin-resistant S. aureus. Also, we could save immunocompromised rats from death. What is also super important for what we're talking about is that you can use reduced time of antibiotic treatment. Eventually, you can also start to reduce the concentration of the treatment, and then this is very important for what we're talking about today. We are currently trying to put together a clinical trial to prove that this can be used in clinic. Okay, so the take-home message number 2 would be that you can reduce biofilm tolerance by using molecules that are weakening the biofilm cells or the biofilm structure. And as I told you, there is also a lot of molecules that have been developed that target persisters or potentiate the antibiotic action. So this is where we are today about what are the strategies to fight biofilm infection in human, okay? So I won't go through all this because we don't have the time, but I just want to point one or two things. I will come back to that, but you see that you can actually, there are different types of a strategy that you can put at different level of the biofilm formation, initial adhesion, the microcolony development, for example, and there are molecules that, for example, are jamming communication between the bacteria that has been shown to be very important to build the biofilm. You can also promote the dispersal, and then there are other, like killing the persisters, that are here at this maturation step. So the question that comes right after is, can we adapt this strategy to treat infectious disease in animals? Okay? Before we ask this question, there is another question that we should ask, that is there any biofilm-related infection in animals and in cattle in particular? So if you just look at a PubMed, classically, looking at the keywords biofilm and veterinary medicine, you'll find something like almost 700 hits. And you'll see that the number of papers where these two words are present are increasing over time. If you look for biofilm and cattle, you'll find something like 600 hits. Okay? So basically, yes, probably there are a lot of infectious disease in cattle or other animals that are due to biofilm. And if you try to get an idea of what are those diseases, in fact, in cattle specifically, you'll find this. So there are multiple ones. You're facing, probably some of you are facing this disease regularly when they are doing your job. And some are very problematic, like endocarditis, pneumonia, problem within the intestine, osteomyelitis, urinary tract infections. They are basically more or less the same disease as in human, okay? And something like that is probably the most problematic, and in terms of cost as well, the most with high cost is the mastitis, so bovine mastitis, that is due to various bugs, but including mainly Staph aureus, coagulase negative staph, Streptococcus agalactiae, and E. coli. Okay, so if we take now this strategy that has been developed in clinic in human, and then if we try to find in the literature some strategy that have been now applied to veterinary medicine, you find a lot, okay? And you find a lot, and especially these last 2-3 years, there's been a lot of strategy that have been developed for human that are now used in the clinic for the vet clinic. And I will show you 2 or 3 very rapidly. For example, for the case of adhesion, initial adhesion, a molecule that is called chitosan has been used a lot in clinic in human, and then now can be mixed with, for example, cloxacillin, and that would be very efficient, again, as a chronic mastitis coagulase negative staph that have been. And this is very recent study of 2018. Another example is vaccination. That is also a very heavy hot topic, I think, in the vet medicine. And then recently, so vaccines, again, E. coli that use different antigens that are related to fimbriae at the cell surface of E. coli have been developed and very efficient to avoid infections by E. coli in case of mastitis. And one thing that is super important, and then that you will face that, and probably this is one of the most sexiest ways, I would say, to kill now biofilm and cure infections is phage. Okay, so phage, they has been for long time completely removed from the market somehow because of the antibiotic. They have been discovered their activity has been discovered in the early 20s by Félix Derel, and then just after that the antibiotic arrives and the phage has been eradicated in terms of strategies, except in like East European countries, like in Georgia, for example, where they are used since now 70 years, in the clinic, in human, daily, and they are very, very efficient. And so you'll see soon that, and maybe already, and there are cases where bacteriophage can be used to cure infections. And then I indeed find one example. Yeah, I did find one example here recently of a cocktail of phage that has been used to clear an infection caused by a SRS that causes mastitis. So there is really, a big hope that this technology then will be rapidly applied into the vet clinic. So the last take-home message I would leave you for is that if we talk about biofilm-related infection, you will find a lot of homology between what is happening in human and what is happening in animals, especially in cattle. That the biofilm, anti-biofilm strategy that have been developed at different stages of the biofilm that are developed in clinic for human are also available for the treatment of animals. And that more importantly, what we are speaking today about is IMR. So we have to put in front rather strategy that will use non-antibiotic strategies that will eventually reduce the concentration of antibiotic that we should use and that will reduce the time of treatment. So finally, I hope I've convinced you that fighting biofilms may help reducing emergence of antibiotic resistance, and that in the future we will use a lot of new technologies to fight biofilms also in the vet clinic. So I will finish just to acknowledge people that have been doing this work, that are these people from the group, Jean-Marc, that is the boss of the unit, and different people that have developed the tools, and then our collaborators for the in vivo model and for the chemistry. And I thank you all for your attention, and I will be happy to answer any question if you have some. Thank you very much.